Hi, I'm John Fowler. I'm a professor in the Botany and Plant Pathology department, and this image is from a couple of summers ago, and it shows me doing one of my favorite things, which is working with maize or corn plants in the field, setting up for pollinations. And we're doing those pollinations to try and understand these small organisms, which are pollen grains from the corn plant. And in this sequence, they're on a microscope slide growing in vitro. This is about the first 30 minutes after plating in media. And you can see the pollen tubes growing out of those pollen grains. And of course, the role of pollen is to transport sperm cells to the female flower to enable production of the next generation. We're interested in the genetics, molecular, and cell biology of these pollen grains, the male gametophyte. And this next sequence gives you an idea of the type of cellular events that we are studying. Here, the focus is on the single pore on the right, through which the pollen tube will germinate out of the pollen grain. Preceding that germination, you see a lot of activity inside the grain, and then you see the cell inside has expanded out through this pore to generate the pollen tube. When placed on a female flower, this pollen tube will then grow through that flower to transport those sperm cells down to the embryo sac and the egg. So as you can see, we do a fair amount of microscopy in the lab, but we have another important piece of equipment that lets us use digital imaging to analyze the products of successful pollination, and that is kernels on an ear. I'll let Zuzana Velipkova, the senior faculty research assistant in my group, tell you about that.
Here we have a corn scanner machine that had been designed and assembled by our previous grad student, Cedar. And it's a sort of a corn rotisserie that allows us to take a video of all the seeds on the corn ear. And then from the video, we get a flattened image of all the seeds on the cob. And we can do all sorts of counts and data analysis from that.
In addition to designing and building the scanner, Cedar, or I should say Dr. Warman now, wrote the software that enables the video to be changed into a projection of the entire surface of the ear with every kernel shown. Because we use a set of mutations that are marked with the green fluorescent protein with the correct lighting and filter, the projections allow us to easily quantitate the effects of each mutation on pollen. Mutations that reduced the pollen tube growth rate, for example, would generate ears where the green fluorescent kernels are present at reduced ratios relative to the expected Mendelian ratio of 1:1. This is Cedar in the field from a couple of years ago. And of course, this past summer, we were all masked up and taking precautions during the pandemic. That's Susanna on the left and Cynthia Waite, an undergraduate researcher, in the middle. Our work this past summer generated a large harvest, which we're busy working on with this scanner. And once the pandemic's over, if you're ever in Corvallis and want to see some cool glowing corn kernels, please feel free to stop by. We love to show off the corn.